Hey everyone, I'm Dr. Masenkai. This is Dr. Gokten.
Hi.
All right, we're going to be talking to you guys today about neuromuscular disorders. Here's the objective slide. We'll have this for you guys. You guys can read this on your own, so we won't read through all these as part of this video. But let's start talking about what we'll be doing today. We're going to go through the peripheral nervous system. The way that I think of it is to think through it by levels. So we'll be starting with the muscles, and the neuromuscular junction, peripheral nerve, and then talking about the motor neurons. In anatomy, you guys talked about the nerve roots, so C6, C7, C8, so we'll skip that today. So let's first start by talking about muscle. So let's talk a little bit about, I guess we'll talk about normal muscle histology first.
Skeletal muscle, of course, when we refer to muscle, we're referring to the skeletal muscle, not cardiac muscle or smooth muscle. And here we have two major fiber types. As you can see, they have some different qualities based on their content. For instance, type 2 fibers are fast myosin type fibers that are involved in explosive speed strength type of activities, whereas type 1 fibers are more slow myosin type with mitochondria rich, high endurance type of. There are additional features to them reflected on their staining patterns, as we will see soon. This is a skeletal muscle on cross section, microscopic view. So as you can imagine, each muscle cell we refer to here is a long tubular structure starting from the origin of the muscle all the way to the insertion. And we're looking at one level on cross section, and you can see they're in polygonal shapes with very little, if any, fibrovascular connective tissue in between, which we call endomysium. That will be important. And then surrounding the entire fascicle will be a perimysium, and that will be important too. And also note that their nuclei are located at the periphery of the myofiber, this tubular structure. There are literally hundreds of them, but in a given section we see just a few of them. So their location will also be important. We will talk about these features. Here is I wouldn't worry about what is dark, what is light, but simply based on the pH you use while doing these stains, it highlights one fiber type one color and the other color. And the point here is that the fiber types are distributed in a checkerboard pattern. So changes to this pattern will also be important in our interpretation. And I realize that not very many of you will become pathologists or neuropathologists for that matter. However, I think it is important to become familiar with how our bodies work and be able to translate that back to your clinical practice so that you can understand the pathophysiology of things.
All right, so let's talk about some muscle diseases. So we're going to talk about a few categories today: muscular dystrophies, genetic myopathies, inflammatory myopathies. We probably won't go much into metabolic myopathies. That's a very complex topic that's sometimes beyond even my scope of knowledge. But some basic things to know about muscle diseases: I think of a progressive weakness. Usually we say it's a proximal weakness, which means the shoulders, the hips, and also the core muscles too. One thing that you should always do if you're concerned about a muscle disease on a patient is to look at a creatine kinase, a CK. It's an enzyme that's located within the muscle, and so if the muscle breaks down, that CK comes out. I think of it as the candy inside a piñata. So if you break the piñata, the candy comes out. If you break the muscle, the CK comes out, and you can see that as you do a blood test. So that helps me a lot.
Except in this case, we don't want to break the piñata.
No, we don't want to. But if it's broken, we can see evidence of the broken piñata. All right, so I guess we're going to talk about general myopathic changes now in disease states.
When we say myopathy, we are referring to the diseases of the muscle per se. As Dr. Masenkai explained, there are multiple levels we will go through. So we are now focusing on the muscle part of this process. And whenever there is a muscle problem, we call it a myopathy. And there are some general histologic features that direct us to that diagnosis. And here are a list of these. This is a general appearance of a myopathic process. In this particular case, it happens to be muscular dystrophy. We will talk about that briefly as well. But let's focus on what the myopathic changes are. So in general, here we have the myofibers. This is a lower power view of cross-section of the muscle. And we have the endomysium, and we have all of them arranged in fascicles. And we have the perimysium, Now you can see that the fibers are of variable size. There are really large hypertrophic ones to compensate for these atrophic ones, which are rounded and kind of entrapped in this increased endomysial connective tissue. There are basophilic ones that represent regenerating ones because some of them got injured. I don't see degenerating or necrotic fibers in this particular slide, but that would be another example of myopathic change. Note that some normally peripherally located myofiber nuclei now moved into the myofiber. We call them internal nuclei. So these are all myopathic changes. There are some other additional specific changes that we will also refer to. And if you find them on top of these, you can make a diagnosis of that particular disease. But in general, if you see these types of changes, we are talking about primary muscle disease rather than nerve disease, for instance.
So maybe we can break it down into a couple main ideas. I guess one is the cells get messed up. They're different sizes. You have the tiny ones that are broken down and the big ones trying to do all the work.
Right.
And the nuclei, instead of being on the outside, like that first picture we saw, are going to be towards the middle of the cell. And then the other thing is the connective tissue. That first picture, we basically saw none of it.
Correct.
And here it's everywhere.
Everywhere, including the perimysium, also thickened. But see, this is basically scar tissue.
Yeah.
And of course, some are trying to regenerate.
Yeah.
It's an ongoing process.